Okay, well, good afternoon, everyone. It is 3:30, so we'll go ahead and get started. For those who don't know, I'm Sarah Woods, and on behalf of EOL, I am pleased to introduce Dr. Patrick Veris. Dr. Veris studied in the University of Colorado Boulder, earning his PhD in chemistry while conducting research at the National Oceanic and Atmospheric Administration Chemical Science Laboratory, CSL. Dr. Veris spent 2 years at the Max Planck Institute for Chemistry in Mainz, Germany as a postdoctoral researcher, after which he returned to NOAA as a research scientist with the Cooperative Institute for Research in Environmental Sciences. In 2018, Dr. Veris was hired as a federal research chemist in NOAA CSL, where he worked until taking his current position in April 2023 as the group lead of the NSF NCAR EOL's Research Aviation Facility Science and Instrumentation Group. Dr. Veris's research has focused on the development of chemical ionization mass spectrometric measurements, or CIMS techniques, for the measurement of atmospheric gases. In recent years, his work has focused exclusively on the development and deployment of airborne CIMS instrumentation. His research focuses on reactive inorganic species with recent interests that include marine sulfur oxidation, heterogeneous reactions of atmospheric gases and aerosol, and reactive halogen species in their upper troposphere and lower stratosphere. Please join me in welcoming Dr. Veris for his seminar titled Innovation of Instrumentation as a Key Driver of Discovery from Laboratory Investigations to Airborne In-Situ Deployments. Quick reminder for those online, enter your questions in Slido at the bottom of the screen, and we will take those after the seminar. Dr. Veris, welcome.
Thanks, Sarah. Appreciate it. Sarah works with me over at RAF, and there's many faces here who do as well, so please reach out. I'm Patrick Veris. I appreciate the introduction. I just started my NCAR voyage about a year ago. It'll be a year in April. And so most of what I'm going to talk about today has to do with the work that I did largely at NOAA on projects associated with that, but some of that spilled over in my tenure here at NCAR. And then at the end, I'll touch upon a little bit about what we do and kind of where we're going, kind of NCAR-wide a little bit. But what I'll talk about mostly today is sort of how I see science from my perspective, which is largely focused on instrumentation. So I'm an instrumentation guy. I developed instrumentation both for lab work and airborne research. And then the science I do with that is kind of the icing on the cake for me. So it all starts with the instrumentation. So I'm not going to give you a huge introduction up front about the science. That will come as we go through this. But what I will do is jump from a short bit to science straight into instrumentation and tell you a little story about how we used it on some airborne missions. So this is my introduction slide. It's very generic, very general. This is a nice figure that I sort of cut up and put some pictures on. What we're doing as a field in atmospheric sciences is really trying to anticipate and identify problems. And we do that on a global scale. So this is a nice figure NOAA CSL designed. In that shows a lot of different sources of contributions to the atmosphere. Volcanoes, oceans, aircrafts, urban cities. These are all inputs into the system, and trying to understand and track how that perturbs the system, what is the chemistry, what are the impacts of that. That's essentially what most of us are doing in the field of atmospheric geosciences. The problem with the Earth as a system is it's very dynamic and complex. And I've got those time series on the right, or vertical profiles rather, on the right, of water, temperature, and pressure. And from the surface to the stratosphere, these are hugely dynamically varying. And that presents a challenge to us. So we need to be able to measure in all of those conditions. And from an aircraft, we need to do that with one instrument from surface to stratosphere often. And so that presents an analytical challenge, presents an instrumentation challenge. And that's the kind of challenge that I like to put myself in the middle of and develop instrumentation to meet these needs. So this talk's really on leveraging state-of-the-art instrumentation to address these complex questions in an increasingly complex atmospheric system. So this is overwhelming for most of you. I'm a mass spectrometrist. This is my bread and butter, so I have one slide that's very complex on it because I want you to just get the basics of chemical ionization mass spectrometry. So chemical ionization mass spectrometry is a technique where we take a charged ion. In this case, I'm showing it for iodide, which is what I've made most of my career doing. And you take an iodide and you cluster it with a sample gas that you sample from the atmosphere or the lab, wherever you are, and you create a cluster. That cluster is created in what is called an IMR, ion molecule region. That goes and gets transmitted down into a detector. Essentially, in this case, I'm showing a time-of-flight mass spectrometer. And this little figure here, this animation by TOF shows what happens once you enter the time-of-flight mass spectrometer. You pulse down, you come back and hit a detector, and you're separating things by mass. So really heavy things fly slower than really light things. And so what we get is a mass spectra that looks like this bottom left figure here. Each one of these vertical lines here represents at least one peak or one gas that we're detecting. And so the point I want to get across here is that we are seeing hundreds of gases in this sample, in this instance, in ambient air. I've called out a few of these. So we're seeing the iodide and iodide water that are created from our controlled formation of iodide, which is our reactive ion. We have a cluster with formic acid, one of the species we measure in the atmosphere. We have nitric acid. There's lactic acid here over on the far right. And then I pulled this inset out of mass 201, so that's nominal mass 201. These time-of-flight detectors are really incredible because they can separate masses at a very high level. And so what I'm showing here is on one nominal mass, we're actually detecting 3 species. One of the more important ones that I'll talk about in my talk here is chlorine. So you can see this little shoulder off to the left. And so that's detection of the 37 mass isotope of chlorine. So these are great instruments. We can measure lots of things at once. There are limitations to it. One of those is that what we get from this is a mass, and that mass is so exact that we can get a molecular formula from that, but that molecular formula tells us nothing about the molecule. And that's one of the challenges of this technique, and that's one of the things that I'll briefly talk about going forward. So it's a very powerful technique. Goes back to kind of the late 90s, early to late 90s. We have lots of different ions we can choose from. Choosing the ion tells us what gases we're able to measure with that ion. So in the bottom of this panel, there's a generic graph of oxidation state or volatility. The H3O-CIMS is one of the older techniques. It measures VOCs, things like isoprene, acetone. As you move to the right in that figure, you get to less and less volatile species. And by the time you get to nitrate ion CIMS, you get things like sulfuric acid. These instruments have been used for a long time in the lab. Aircraft work started somewhere in the late 90s with a lot of the folks at NOAA. Greg Huey, who is a good friend of ours here, Lee Malden and others had a big hand in this. And so I'm going to talk specifically in this talk, though, about iodide CIMS, which is what a lot of my current work has been focused on. So brief history, because this is the foundation of what I'm talking about here, is how do we get from a technique into an airborne instrument? Why does that matter? And what is that process? So sometime in the early 2000s, iodide CIMS was almost exclusively used to measure these peroxyacetyl nitrate compounds. These are organic compounds that capture NOx and can transport it long distances from urban regions. Very interesting science, and where, in my opinion, iodide CIMS began. Very rapidly, we found out that iodide CIMS could measure the species ClNO2. It made some waves in the community. It's a very photolabile species that activates chlorine in the atmosphere. And so it started to see mass use as a detector for more species than PANS, and ClNO2 being one of them. By the 2010s, there was a move from quadrupole mass spectrometers, which could really only do one mass at a time, to the time-of-flight analyzers that I just introduced. This was a huge advancement because now we can measure hundreds of molecules at once on a sub-1 Hertz time scale. If you saw Doug Warsnap's talk here last week, he talked about how he coined the phrase the molecular revolution. And it's true, we have hit a molecular revolution where we're now seeing species in the hundreds rather than in the dozens. And so this is a really cool time to be part of this field. The figure here shown is the progress of resolution from quadrupoles up to where we are now with time of flights. So the original instruments back in 2004 or so, you would have a spectra that looks like this yellow line, very broad. That spectra, in this case, includes about 5 different species, and we could not tell the difference between those. It's one of the limitations of the instrumentation, and it's one of the reasons why we have to think carefully about old data sets from the CIMS instrumentation. As time went on, these got better, they got longer, they got higher resolution, and we can start to pull out individual isomers of the same molecular formula. So I'm going to talk in the next couple slides about what I'm going to call mundane work. It is trying to understand the technique, trying to understand the sensitivities. Mundane work is interesting. There's this really nice Ed Helms, he's a comedian. I think you would know him if you saw him. He said there's something beautiful in the mundane if you take enough microscope to it and focus on something that seems innocuous to begin with. The idea of studying the mundane in instrumentation is really what's guided my entire career. It wasn't the science question, it was the instrumentation. What can we do with it? How do we make it better? And what can we go out and see? And so that's sort of the underlying principle of this talk, and it led to most of the science that I'll get into. So studies of mundane part 1. I'm going to go through this stuff pretty fast because it's not that interesting to an outside observer. It's not exactly science-y, but to me and to instrumentation people, this is really interesting stuff. So key step in CIMS work, I just said you only get a molecular formula, and that's a limitation. We want to go out there and we want to see what formic acid concentrations are. We want to go out there and measure some new alpha-pinene oxidation product. And so what we need to do in the laboratory is take the data sets that we get, identify peaks, and then build sources in the laboratory to verify those measurements. We have to quantify the detector, the sensitivity response to those molecules. And one other thing that I think is underplayed in the field of CIMS and calibration is that these sources, this work, really helps us to identify potential limitations in the use of these techniques. Calibrations, when you put them in your instrument, you might get fragmentation. You might see something you don't expect. You might not even see it at all, and it might be something else that you're seeing. And so this is how we fine-tune our observations, fine-tune our conclusions, and it played a huge role in the science that I'm going to talk about. These are just 4 examples going from left to right, earlier work to current work. On the far left is back when we were using iodide for almost exclusively pans. I worked with Jim Roberts to develop some calibration sources for pans, work with some folks up in Toronto to do some HONO calibrations. I'll talk about a little later this next one. Most of my talk will be on this molecule, hydroperoxy methylthioformate, HPMTF, it's a mouthful. I'll get to that bit later, but that's a really nice calibration setup we did that we also got some kinetics out of. And on the far right, some work from Mike Robinson, who's currently at NOAA Chemical Sciences Laboratory, and he's been doing a lot of work synthesizing organic nitrate species. And he'll be given a thesis here up in April 2024. It's a really nice body of work. I encourage you to reach out to him if you have interest in organic nitrates. Part 2 of the mundane is the responses of the instrument to flying around an aircraft. So now we have a calibrated instrument, we know what we're looking at, we put it on a plane, we fly it around. So I'm going to draw your attention again to that figure I showed on slide 2. The dynamic range of water, temperature, and pressure are huge in the atmosphere. And no matter what instrument you put on an aircraft, it's going to have a problem with one or multiple ones of those. In our case, it has a problem with all 3. And so we have to really understand how the instrument behaves, and ideally what we want to do is isolate the instrument from those responses. And so on the very top here, on the left, I'm just showing our sensitivity, or just detected signal, versus ambient pressure, or IMR pressure in this case, which is pressure of our reaction region. And as we increase the pressure, the signal goes up. And that's happening because the reaction that's controlling our detection is a collision of an iodide ion plus our sample gas. So as you go up in pressure, you get more collisions and you get more sample. So the only way to control this in an aircraft world, in our experience, is to put a pressure-controlled region on the front end. I'm not going to go into what that looks like, how we design that. I will just say that we have to create a region front that separates the aircraft, the outside of the aircraft from the instrument itself, and we've successfully done that over the years. Temperature is the next one. Again, Mike Robinson, he has a really nice paper in AMT that I think is exemplary for how we study temperature in the system, what we can learn from it, and what kind of details about sensitivity we garner from that research. It's not interesting if you don't like iodide CIMS. It's very interesting if you use iodide CIMS, if you want to do CIMS in the future. So what happens in this situation is the reaction to form this cluster. You now have a cluster in your system. As you increase the energy in that cluster, it'll fall apart and go in the reverse direction. And so you end up losing signal with temperature for some species. You can see huge variability between that. And then the last one's water sensitivity. So we fly around in the atmosphere, we get very large differences in water from the marine boundary layer to the tropopause. Those can impact a sensitivity change in this instrument. In our case, it relies on this formation of iodide plus water cluster. As we change the water, we push that towards iodide water. We can also react not only with the I-, but we can react with the iodide water, so we get huge changes in sensitivity potentially occurring in the system. And so During ATOM, which I'll talk about later, it was a NASA mission. In between two sections of the mission, I worked with a colleague, Andy Newman, to develop this dynamic water addition that helped to control the humidity of the system and our sensitivity to it. So these challenges, I went through that very quickly, but they're foundational to understanding what the instrument's doing once we get in the field. And there's a subtlety in the observation that I'll talk about for HPMTF that relied on both the calibration and our understanding of the instrumentation to even be able to see in the first place. So we throw that all together, we have a beautiful instrument for aircraft research. We have an inlet coming out of the plane. Here's a nice picture of inlets off the side of the NASA DC-8. Inside that, we can add cal gases, we can add zeros, the details of which I won't go into. We have inlet flow control, we have inlet pressure control, we have water control in our system. We have all the things that'll isolate the actual instrumentation from the outside of the aircraft. So we're ready to go do a project. This is what the instrument that the bulk of my talk will be on looks like installed in the NASA DC-8. It's an iodide TOF-CIMS, which I've just kind of built up to. So the atmospheric tomography mission. This is a really fascinating project that was done from 2016 to 2018. It's a NASA-funded EV proposal on the DC-8. We flew around the world 4 times during those years. 2 of those years, the ATOM-3 and ATOM-4 deployment, this iodide CIMS was part of the payload. The instrumentation has sub-PPD detection limits, 1 hertz frequency, so we get really great measurements of a lot of species. This little table inset just shows a subset of the species we can detect. That's what we actually reported on the mission, although we can provide dozens of organics that we haven't calibrated for. This is a lot of work. So the data is published, you can find that there. The figure there from a BAMS paper in 2022 is a really nice graphic that shows how we flew ATOM. So these were fixed flights around the globe, 4 flight tracks shown in the upper right here. Going from surface about 500 feet up to tropopause and back down. And so we did this all the way across the globe, getting vertical profiles. The idea was to fill in gaps in modeling observations available for constraining models. So really, really unique project, really amazing to be part of. The iodide CIMS deployment yielded some really great results from this. Some of those results were expected, like halogen observations. We knew going into this, that's one of the goals that we had, was we could measure chlorine, we could measure BRO, we could measure BRCL, and we wanted to get observations of that. Top right panel here shows sort of a vertical profile separated by region, so Arctic here, North Pacific, Tropic Pacific, South Pacific, and Southern Ocean. So these are vertical profiles of chlorine. They're colored by ozone. You can see some stratospheric halogens coming down, mixing into the tropopause. The rest of that's just marine halogen chemistry and some other interesting features. Shown a different way, here's a curtain plot on a projection of the globe. So we're doing vertical profiling, and the colors are a heat map of where chlorine is showing up in the atmosphere. And so we have some really interesting features there. A lot of this is summarized in this Wang 2021 paper that takes a bunch of halogen data sets and says, what do we really understand about halogens? We also look at formic acid in the remote atmosphere. That's a really interesting study, but we expected to see formic acid. That was one of the things we went in knowing that we were going to study. And then there were unexpected discoveries. One that just came out, Jim Roberts, who's in the crowd here. If you're interested in this, you can ask him about it afterwards. We detected BRCN. It was a species that we hadn't seen before in the atmosphere. It's really interesting. It's connected to ozone depletion events in the Arctic, showing really nice correlation, anti-correlation across the entire Arctic swath. So that was one of the unanticipated discoveries we made because of the detector was so accurate and measured so many things at once. We could kind of dive in and find things we don't expect. We repeated this a little bit again, and this is what I'll talk about for the remainder of the talk mostly, are global observations of a new species from dimethyl sulfide oxidation. So this is where I'll focus on the next few slides until I get to jumping back into the stratosphere for just a little bit. So this is where I'm going to jump back into science. So why am I going to talk about dimethyl sulfide? I feel like I have to introduce that in order to tell you a story about it. So marine phytoplankton. So on the first slide, I had some nice pictures of phytoplankton. This is why. Phytoplankton produce this dimethyl sulfonium propionate in the ocean. That will turn into dimethyl sulfide in the ocean and high enough concentrations that it partitions out of the ocean into the atmosphere. It's one of the largest sources of sulfur. Up until recently, it was always talked about as the largest natural source of sulfur to the marine atmosphere. Some new work from Becky Alexander and University of Washington is showing that maybe passive volcanoes are the largest source, but nonetheless, it's a very important part of the remote atmosphere. For about 30 years, we've been studying this. This cycle here over on the left, it was published in 2011 by Quinn and Bates in Nature, and it's a sort of critique on a cycle that we've called the claw hypothesis, which is the idea that when phytoplankton produce this dimethyl sulfide that comes out of the ocean, you can have chemistry that occurs that converts that to sulfate. That sulfate can then serve as cloud condensation nuclei and create clouds, and those clouds can reflect sunlight. And that creates an albedo effect that can either increase temperature or reduce temperature, which then feeds back into the production of phytoplankton in the ocean. And now you have a cycle that occurs. So we've been studying that since 1987, when this paper came out in Nature. And 2011 was the latest sort of statement on it that basically said the effects aren't strong enough to have a coupling effect and sort of broke the hypothesis. But the point here is that we've been studying this as a climatically relevant system for over 30 years now. And up until about 2017, this generic schematic is what that world looked like. So we had DMS come out of the ocean, could do a couple things, but it would ultimately form sulfate. And then that sulfate was part of a system of CCN and particles that is formed over the ocean from bubble bursting, sea spray. All of those have relative contributions that go into cloud formation. Those clouds can then reflect sunlight. And so we had this general simplistic understanding of how DMS fit into this scheme. And I really like these figures on the bottom here. One was published in 98, the other one in 21, that show pretty much the same thing. It shows how little things have changed, but it shows how much we've changed in graphic design. So I really like it. We're still talking about the same stuff 30 years later. We're still talking about how clouds are pumping air up through cloud systems, how particles are coming out of the top, being formed at the top. How do these things feed into that system and form new particles? And so I'm going to dive into these processes and how we've changed our understanding of the system through deploying this new instrumentation. So we went out, so the paper I highlighted, we went out and we discovered a new DMS oxidation product, HPMTF. So I'm showing chemistry here. Some of the little chemistry I'll show. I am a chemist, but I'm trying to take it easy on the crowd of non-chemists here. So dimethyl sulfide will react with OH in the atmosphere and form a peroxy radical. That will react on itself twice and form HPMTF. The chemistry was theoretically known at the time, although we were unaware that it was theoretically known, that this should occur in the atmosphere. And not only should it occur, but if OH reacts with DMS to remove a hydrogen, almost all of that should form HPMTF. So this is a really important pathway in the atmosphere. Not only did we see it for the first time, but we also all of a sudden had a global map of concentrations of HPMTF in lots of different conditions. That's how amazing this ATOM project was. And the take home here is that we change from this traditional viewpoint of chemistry where DMS oxidation will essentially form two things, SO2 and MSA, and almost half that, more than half that SO2 is actually going to form HPMTF directly from DMS. And so that's a big impact on our understanding of that system and how sulfur moves through it. This part of the paper here is, I think, understated. I think we have very little ability to look into new particle formation events. So in the atmosphere, when you get really clean conditions, you don't have a condensational sink, and you form a really low volatility gas. It can self-nucleate and form new particles in the atmosphere. One of the molecules that it does that through is sulfuric acid. Those measurements are very, very difficult, especially from aircraft. So it's rare to actually see the process, A, from an aircraft, and B, see some of the constituents that are involved in that process. This graph here shows the process I'm talking about, where you take a molecule like sulfuric acid. It can nucleate into what we call the nucleation mode of particles. Those can then start to coagulate. Things can condense on them. We can kind of go from the Aiken mode to the accumulation mode and onward up to form particles that might turn into clouds. And so this process is understood. It's well known. When we went out on ATOM, we saw the process from nucleation up through accumulation mode. Here I'm showing particle number concentrations versus time. And we see I've got these little slices here showing you the correlation to the size modes that are in this graphic here. So the very top band you see is the accumulation mode. These are larger particles. The middle band here is the Aiken mode. And the lower end of this graph is the nucleation mode. And if we look at a time series of that, so light blue to light blue down here, and now the black here is HPMTF, we see really good correlation between when we see new particles in the atmosphere and HPMTF. And this is drawing a relation between the DMS oxidized products and the nucleation particles that are formed in the atmosphere. And so this is a really great way to directly observe some of these events. This is a really unique data set, and I think it's something that we should go out and try to do a bit more of, is use measurements like HPMTF to follow the chemistry that is forming these particles. I'm going to jump back to measurements for a second here, because the story on how we found this thing is the reason why I think the instrumentation is so important. So HPMTF was hiding under our noses the whole time. So 30 years of science, 30 years of measurements, it was there the whole time, and we just didn't realize it. So going back to the quadrupole instruments down in your bottom left, we get these nice broad peaks that encompass a whole lot of stuff. And one of the goals during ATOM for us was to measure the species N2O5. And so N2O5 is a nighttime species. It can form nitric acid and ClNO2. Let's not update right now. And so during the daytime, N2O5 is very thermally labile. It should thermally decompose into NO3 and NO2. And so we shouldn't see it during the daytime. So we're flying over the ocean at about 500 feet in the middle of the day, and we start to see what looks like N2O5. Very clearly, the science just tells us that can't be there. It's not real. So now we have to sit there and decide, what is it? Do we know enough? Does the instrument tell us enough to figure out what it is? So the understanding of our stability of the instrument, some of the changes we made between ATOM 3 and 4 to the water control in the system, things like that allowed us to look at the mass spectra in such accuracy that we could determine the difference between a subtle shift and a peak, where N2O5 here in the upper troposphere is the black peak here, and the green is this HPMTF product. The shift in mass that we're seeing there is 0.0074. The instrument should really only be able to measure to about 0.02. But because we have such a stable instrument, we have such a profound understanding of the technique due to all the development that we've done, we can have confidence saying this is a real shift and a peak, and this is a real molecule, and enough confidence to follow that down the rabbit hole for about 2 years. So in those 2 years, what we did is we worked to develop a laboratory calibration system that we also could then leverage to do kinetics experiments. This is work that's mostly done by Emmanuel Asaf and Zach Feinwax in Jim Berkler's lab at NOAA. We designed this thing to give us a calibration for HPMTF, but also provide an identity. So again, we have a molecular formula. We don't know much about that. We have a theory. We have to test that with the laboratory calibration. So we put a lot of work into this. This is a movable sampling apparatus where we can photolyze chlorine molecules, react them with DMS, and then we can sample at the various points along this reactor to give us a reaction time scale. So we put a ton of work into this. I think I can't understate how much work is on this slide right here. These two guys and myself put about 2 years of work to generate this figure on the right here. The theories from the original work that we did in the ATOM paper, and these blue data points are the measurements of the rate constant. So what we're measuring here is the reaction rate of DMS to form HPMTF, which is the first direct measurement of that rate. This also provided a really nice calibration for the work. The next thing that happened as part of the ATOM or follow-up to ATOM was that we deployed the same instrument, the iodide CIMS that I showed you before that was on ATOM. We deployed this during the FireX project. And as part of the FireX project, there was the NASA SARP program, the Student Airborne Research Program. And it provided us with a flight of opportunity where they basically gave us one flight to go into the marine atmosphere and figure out if we can extend the story a little bit. And what I really like about the story, besides the science being really incredible, is that this is representing about 1 hour of flight time that this PNS paper came from. And it's so much information that we learned from this 1 to 2 hour flight. And so it just shows you the power of airborne research as a whole. We went out, we took the instrument out again, we flew over these cloud streets, and so this is a nice little generic picture of this. Most of this work I want to point out is Gordon Novak, who's now at Chemical Science Laboratory in NOAA, but at the time he was a graduate student at University of Wisconsin. So Andy Newman and I took the instrument out, collected the data. Gordon did this nice analysis. And what happens is as you fly underneath these clouds, you get updrafts into the cloud, you get downdrafts out of the cloud. We can correlate that with vertical wind speeds in in black, and solar radiance in blue. So when we're in a cloud gap, you get more sunlight and you get a downdraft. And when you're under cloud, you get an updraft and less sunlight. And then you can take the wind speed, so again, vertical wind speed in black, and correlate that with HPMTF observations in red, and you get something that just looks like you shouldn't believe what we've done there. But what it's showing is that as you go into the cloud, you're removing HPMTF. As you come out of the cloud, you have less HPMTF. And so you can take those observations and we can actually convert that into a flux into clouds, or loss rate of HPMTF to clouds. And also, by chance, is an approximation of the entrainment rate into a cloud, which is a really interesting observation. We can look at that on a global scale and say, in a global model, and say, what is the fate of the DMS sulfur when we put this into models? And you can see this big, large blue chunk is the cloud uptake of HPMTF. So it's showing us that across the globe, one of the most significant sinks of DMS sulfur is due to cloud loss of HPMTF. When you put that into a global model, you get the result on the right here, which shows very large changes in SO2 and sulfate as a result of this additional sink of sulfur to clouds. And so this has pretty big impacts on the climate, impacts of the DMS sulfur system, and will alter how we understand that claw feedback loop and some of the chemistry that we talked about earlier in this talk. So from 2020 to today, we've done a really remarkable job as a community of filling in the gaps of our understanding. So in the center of this, I have HPMTF, really interesting stuff, and all the pathways it can go. We've learned a lot about how the system fits together, but we still have a ton of questions that remain. We have questions about, do we really totally understand the budget of sulfur? There's a new emission that's been published on this from Novak et al. From the ocean. So methane thiol comes out of the ocean and it can form SO2. So here we've removed an SO2 source, and then we more recently put one back in the box. And so how do we understand this? Do we understand this? Is there stuff that we just don't see? And one of the limitations of this sort of research in the remote marine atmosphere is we just don't have a ton of data on it. And that's where the aircraft work comes in. And so Drew Rollins and I, with all these questions on the right, what's reactive nitrogen doing out there? What kind of air-sea exchange do we not understand? We got together and we designed a project to do some more airborne sampling, and we called that Aroma. We paired with a set of researchers to do an urban study, but the idea of this study was that it would give us both time to do marine research and urban air quality research on the NASA DC-8. So this was a NOAA project that we conceived somewhere 2019, and it got delayed by COVID, like most things did. And so by 2023, we were ready to finally carry out this mission. We had a really great set of people that worked with Drew and I to do the planning for the flights, do science tracking, come up with a traceability matrix for the project, and then be in the field to sort of direct the plane around in real time as we're setting cloud systems and the complexities that go with that. The map here in the middle shows the flights that we did. In orange, those are the marine flights that was part of Aroma. The black was the urban side of Aroma. If you're interested in that, you can talk to Becky Schwantes or Carson Farnicki at NOAA about the urban side and what they are starting to understand about urban chemistry. I'm going to talk exclusively about the orange flights there, which are the Aroma flights. The little video down here shows the kind of conditions that we put ourselves in and the banking of the DCA. That was pretty nice. We're trying to get in the clouds. It's a space that in-situ gas phase people and aerosol people don't like to work in. There's a lot of problems with getting in and near clouds. But now with the technology we have, with the instrumentation we have, we can start to really understand when we're getting valuable science and when we're not. And so we decided to go just launch in, get into the clouds. We did that with a really comprehensive payload. This is the DC-8, schematic cartoon of it, and all the instrumentation that we filled it with for the Aroma project. It was a nice mixture of things targeting urban and marine chemistry. On the bottom there, because I'm a CIMS guy and I'm highlighting CIMS technology and the impacts, I'm highlighting that we had 4 CIMS instruments on this payload. That's a really big chunk of the payload to dedicate to one type of instrument. The reason you would do something like that is that because we get so many species measured with these processes, and they span such a wide range of volatility space that we get a ton of information from them. So we had an iodide CIMS, a CF3O- CIMS, ammonia CIMS, and a PTR H3O plus CIMS. Down there on the right is a picture of the CF3O- CIMS by Caltech, John Krauntz, many of you know him. So we went out and did these flights. Here are the 4 flights colored in the little mini map here. 4 separate flights spanning from about 24 north to 43 north. We tried to put ourselves in very different cloud fields throughout these flights. I'll show you a couple of these videos here. I think it's important to look at these in video form because the clouds are dynamically moving, the plane's dynamically moving, and so it really matters whether we're sampling in cloud-free or cloudy conditions and what type of cloud conditions we're sampling in. So in this particular flight, we headed north, we did some clear air sampling, and then we headed due west underneath this cumulus cloud deck. We're looking for how the clouds are impacting that clear sky chemistry. We're looking for how the distribution of sulfur species changes. We want to do different maneuvers to get eddy covariance flux measurements, things like that. So I'll show you some more flights as we go on. We did some ship plume sampling. We did some sampling in cloud outflow regions, trying to look for that new particle formation I highlighted before, trying to look at emissions coming out of the cloud and how those oxidize above cloud. The figure on the far right is a little animation that's showing data below 1,000 feet in black, and then it's going to flip and it's going to show you colored by DMS. I have to show you the black because it's over the chlorophyll in the ocean, and when it switches to the color of DMS, you kind of lose the data points. In most cases, except for some outliers, like this one down in the south here. We're trying to understand how you can relate the activity of the phytoplankton, which is represented by the chlorophyll, on the DMS gas phase emissions and concentrations. And so this is some really neat work that we're going to continue to do. What are the dependencies of that? Wind speed, how do clouds impact that? How does temperature impact that? And so this is just the beginning of looking at these types of data sets. We did, like I said, coastal surveys. So this is another way to look at the data. I've taken a few sort of sections, these white spots, and looked at the sulfur species distribution here. Those sulfur species that we're looking at here represent a very large part of the DMS oxidation scheme. So for a long time, we weren't able to access many of these intermediates or product species from a measurement perspective. Our instrumentation that would measure OCS and SO2 just didn't have detection limits to measure over the open ocean, where concentrations are very small. With the innovation of new instrumentation like the LIF, which Drew Ron's developed at NOAA and is being tried to, they're building it here at ACOM, you can measure SO2 concentrations that are in the PPT range. So now we can start to access sulfur product measurements in this space, and on Aroma we covered 6 of these, which is a very large portion of this whole scheme. So we can start to trace sulfur through the system and understand what impacts the pathways and how do you model this and what are the climactic relevance of those changes. And so this just shows a time series of us going in and out of the boundary layer. When we go into the boundary layer, we see sulfur species that are due to the DMS chemistry. When we leave, you get clean atmosphere above the boundary layer. And so we're gonna try to start to study this and understand the gas phase processing and model this process and look at the dependencies. In addition to that, we had a lot of aerosol and cloud observations. And so I have a list of all these observations here. I'm not going to go into the results here. This is really, really new data. And I want to stress that this is all preliminary data you're seeing here. And so these numbers could change quite a bit. But we have nice measurements of particle sulfate, particle concentration, CCN. This is another flight that I think is of note. We went up north and sampled in some clear sky conditions. And then we saw that this nice little open cell cloud system was forming down south. So we took the plane and directed it down into the system. We do these box maneuvers, which are attempting to get some eddy covariance fluxes. We're trying to do some sulfur mass balance with these boxes from surface to boundary layer height and above. And I've taken that and shown you a little 3D plot of what that looks like with some HPMTF observations on top. So here we're seeing some chemistry that's going on above cloud layer. So as DMS goes through clouds and does not get scrubbed, it comes out the other end and can oxidize. And that's where you get HPMTF formation. And the question's, what happens then? Do we get sulfuric acid formation above cloud that could lead to new particle formation? Do we get uptake of that HPMTF back into the cloud? And so this is a really great example of the type of data you can get with really focused maneuvers in cloud fields with really great instrumentation. We hope that this aroma study, it was only 4 flights again, we're hoping that this motivates a lot more research into remote regions of the ocean, trying to understand the balance of not just sulfur species, but organic carbon, trying to understand how you get particle formation and movement through the boundary layer to the tropopause and how you get new particle formation. All these processes are coupled, and all of these chemistry, all the chemistry feeds into that process. And so we really want to motivate future work into this. All of that was done with this tropospheric CIMS instrument. And I have Andy Newman, who's in the crowd here, his face here, he's now retired. The 2 of us built this instrument together. It's a instrument that's built to go in a cabin. It's user operated. Flying around a plane, if you've done it, is tiring, especially when you're doing 14 flights, 15 flights, whatever. It's a very exhausting process. We can go from surface to about 12 kilometers in the DC-8. We haven't flown out on any other planes at this point. And it doesn't tolerate low pressure environments. If the instrument was in low pressure, it would arc, but the people also can't tolerate that. And so it has its limitations. It's a really powerful tool, as I just showed you, but it has limitations. We then took what we understood about the instrumentation, the developments that we had, and came up with this idea to build this instrument that would go on the WB-57 that would allow us to access data in the stratosphere. And so proposed to build this, and in the last few years of my time at NOAA, with a large team, including many people, engineers, software programmers, scientists, physicists, came up with this stratospheric CIMS instrument. It's, in my opinion, the next generation of CIMS instrumentation. So this is a fully automated CIMS instrument, doesn't need an operator on board, heavily engineered to withstand pressures down to about, well, probably lower than 60 millibars, ambient temperatures at 200 Kelvin, and can operate somewhere from about 10 kilometers up to the highest we've gotten is at 18 kilometers. So we're accessing a really unique part of the atmospheric system. These are the first data that are coming out from this project. I really appreciate Gordon Novak for letting me share this with everybody. He gave a nice poster at AGU that had a lot of this content in it. We're accessing a part of the atmosphere where previously we just didn't have, A, good enough instruments with good enough detection limits to make some of these measurements in situ, and B, the engineering just wasn't there to get them up there. So the NOAA Earth Radiation Budget Project is designed to go study the stratosphere as it exists today in the event that we might start perturbing that or natural perturbations like volcanoes occur. Do we really understand what's happening up in the stratosphere? The CIMS is a great instrument to put up there because, as I showed you, we can not only measure the halogens, which are a really crucial part of the system, but we can measure whatever we want if we change the ion chemistry in it. If we put nitrate in, we can start to measure really low volatility products. So what I have here is Cold Saber, which was a recent project. I was in my last project with NOAA that I was involved in, and it took place in February, March of 23. We flew out of Houston and went up to Fairbanks, Alaska, and did a series of stratospheric flights in Alaska. The data on the bottom left is now coupling the ATOM observations that I showed you earlier from halogens. So this is N2O5 and BRO with the new measurements from SABRE. So we've taken our data set and essentially extended it from our 12-kilometer cutoff that we had during the ATOM DC-8 flights. And now we're getting much higher into the stratosphere where you start to see concentrations of halogens like BRO, Elevate, and N2O5, which is, back to our original point, something we were very interested in to begin with going into ATOM. This system here is what we're really interested in getting at the core of. As we start to think about perturbing the stratosphere, as we start to think about chemistry in the stratosphere, these halogens play a really large role in ozone destruction and propagation, but also in NOx regulation, how nitrogen is transferred through the system, particularly through CLO-NO2. And these are some of the first in-situ observations of CLO-NO2. So here I'm showing altitude versus latitude. So this works, all the stratospheric work out of Fairbanks. So you see up in the lower stratosphere, really high concentrations starting to show up of CLO-NO2 and BRO. This is 500 ppt of CLO-NO3 or CLO-NO2. This is just another way to look at the data of vertical profiles. So this instrument is just really incredible piece of engineering. Very early on in its life, it took about two to 3 years to go from conception to development. A lot of steps involve engineering. It's really the pinnacle, I think, of CIMS airborne instrumentation, and I hope that we can learn a lot from this and keep developing instrumentation like this, because what it's going to allow us to do is study in regions of the world we weren't previously able to get observations in. And I showed you a bunch of data from ATOM that just showed the capacity of these instruments to produce good science, and I expect a lot of good science to come from this. So this was done with a ton of people and a ton of logos. And so I'm going to stop here for a second and just recognize the work of a lot of people that went into the development of these instruments, the science, the projects themselves, the ATOM team, the Aroma team, the Sabre team. A lot of faces are attached to this. A lot of incredible work is attached to this. And so I think we need to recognize that anybody that comes up and gives a talk is probably talking about more work than their own. And that's definitely the case in this particular one. And many of the people I work with are out in the crowd right now. But one thing this whole talk is focused on is that nearly all the work here was done in laboratories, research aircraft, and with financial support provided directly from the government. NOAA, NASA work. And access to those resources is just limited to government research a lot of the times, particularly large aircraft work. And so when I came to NCAR EOL, I thought a lot about what the differences are between what I was doing and what I will be doing. And I think the coolest thing about NCAR, NSF NCAR EOL and RAF is that this is a community resource for everybody to use that gives them access to the types of resources you just can't get in an academic institution. The TOF-CIMS I talked about is about, I'm being sort of conservative here, $600K to buy one commercially. It looks like that. About 2 to 3 years to build it up. You need engineering, you need mechanical, aeronautical, electrical, others. You need parts, inlets, racks, certifications to fly on a plane. You need funding for scientists to work on it, technicians to build it, and you need deployment costs. And these add up to very, very large numbers. You could buy one of these instruments, you could run it in the lab, you can have a whole career doing that, but you can't access that aircraft piece of it. And so what we're doing here at NSF NCAR is really amazing in that it takes all of that, bundles it up, and gets us projects like the NCAR APROC. I didn't ask Brett if I could show this, but it's pretty well known at this point that ACOM, and NCAR ACOM group, is developing a pod-based CIMS, similar to what I just talked about, and I see it kind of as a middle ground between the tropospheric CIMS that was on ATOM and the stratospheric CIMS that we flew during Sabre. Somewhere in the middle, you have APROC, which can fly on the G5 under the wing. Really small inlets. It's going to be a huge technological advancement to what we have. It's going to be community requestable, which is a big deal. And it'll provide some of the science to a broader community than just the folks at NOAA or just the folks at NASA. And so I'm really excited about it. And Brett Palm is really excited about it too. So we're creating access to broader community. How are we doing that? In my opinion, what we are doing and need to do more of is leverage not only our internal expertise, but collaborate across NCAR groups. I'm working with Brett a little bit to develop some ideas for the pod-based CIMS. Kyle Zarzana's back there. We've talked a lot about how to make inlets better, things like that, how to take some of the technology I talked about and adapt it for this. We need to look out in the community and work with folks to take innovations that are happening for ground-based instrumentation, laboratory instrumentation developed from aircraft. And so this is what we're doing at NCAR, specifically RAF, hopefully. And you can't do that without a great team. Many of my team is here. Sarah, you've already seen. Apologies to Stuart. I couldn't find a picture of Stuart, so he's a stick figure up here. I promise he is a real person. This is my team at RSIG, Julie, Mike, Teresa, Stuart, Sarah, and Britt. We're looking for a next member of our team, hopefully to come in and do some aircraft wins. I showed the ATOM, the FIREX data that we looked at, cloud streets, and looked at cloud uptake of HPMTF, really profound study. Couldn't have done that without really great aircraft wins. Everything is important from the most complex instrumentation we put on the plane to the temperature sensors and the wind measurements. And so our group takes care of over about 60 airborne instruments, covering clouds, radiative measurements, aerosols, gases, and beyond. I'm very proud to have a good group of people working very hard to manage that. We're about to start the Caesar project. We're doing test flights this week, hopefully, fingers crossed. And we're gonna go to Sweden, do this cold air outbreak experiment here shown on the right with the C-130. So I just wanted to highlight that. Paquita, Bart, and Greg are the PIs for that. It's gonna be a really great project. So with that, I think I'm done. So I appreciate your attention. I'm happy to take questions, and I give you a ton of information on instruments and science, but we have some time to talk about it. So thanks for the opportunity to talk to you all.
Thanks, Patrick, for that extensive yet approachable seminar. That was really interesting, and I'm sure there's lots of questions. Who would like to start?
Thanks. That was a really good talk. Thank you, Patrick. I'm curious about what the scales are, like what are the vertical and also horizontal scales for heterogeneity when you're dealing with chemistry? Are you know, obviously if you're flying a flight leg, you have to go forward to stay in the air. So how does that impact your sampling strategy and your ability to infer the structure in the atmosphere?
Yeah, it's a good question. I think this. So I really like this figure. It kind of got buried in this paper, but it shows a lot of that structure. And I didn't point out some of these features, but you only get this nucleation when you don't have large particles to condense on. And so if you look in this band up here, you have these gaps. So if those large particles disappear, there's no condensation sink, and now you can form new particles. You know, this red trace is a vertical profile, so we're flying up through a cloud layer to about 2 kilometers. So we're talking about pretty small scales on an aircraft space. And so easy to miss with old instruments. We, in fact, probably did. With averaging instruments, can samples, we are going to miss things like this. And so having fast instrumentation with high resolution is really crucial in a lot of this. In retrospect, if I could have saw that in real time, which I don't know that we had the capability to do at that time, I would have said, stop the plane, we got to stay in this for 4 hours, right? So I think you need good instruments that have high time resolution and just high resolution concentration-wise otherwise. But you also, I think, need to think about a software side of this with aircraft research, which is real-time feedbacks to what you're doing. I know we've talked about HSRL and dial and having some of those real-time feedbacks where in FireX and you can use those to really find fire plumes and fly in them appropriately. Otherwise, you will totally miss that. Thin sheets of air in the Arctic, on ArcPac when you're flying there, that you see them in the distance, but you'll never find them because they're 100 feet deep or something like that. So scale is important. Marine boundary layer, I'm currently looking at chlorine chemistry that's happening in about 200 feet. And so really fine structure in that. Yep.
Thank you.
Thanks for a great talk, Patrick. I have a question about chlorine nitrate. There's some interest, or maybe I should just personalize it. I'm interested in whether there's chlorine nitrate in the lower atmosphere. Did you see any of that anywhere in your travels?
I can't say that we have. I also can't say that the ATOM instrument can measure that because I don't know that we've done it. I'd have to follow up with Mike Robinson, Gordon Novak, and others at NOAA, whether on the tropospheric SIMS they actually see it. This data here in the Strat SIMS has been verified with laboratory calibrations, things like that. So, this is where I think going into Sabre, I would have said we probably won't see this as a cluster with iodide. And then we built an instrument that does see it as a cluster with iodide. So that goes back to kind of really, do you understand your instrument? Can you make a cal? Can you do all those things? So I haven't seen data from the troposphere that I think is real. I don't know that it hasn't happened or doesn't exist.
Yeah, very interesting. You mentioned that the time response of this SIMS is better than a second. What limits the time response? Is there any potential for going a little bit faster?
It's, so this animation here, it's pulsing these ions. Typically, we're doing that at 25,000 times a second. And so we're getting a full mass spectra, in theory, 25,000 times a second. But in those mass spectra, they're mostly blank and empty. And so maybe you get one ion somewhere. And so what you're seeing down in the bottom left is the sum of all of those spectra on top of each other. And so you're limited by signal. So if we're somewhere like a fire plume where we have a lot of signal, we might be able to operate this thing at 20 hertz. and get enough signal to measure something. If we're out in the remote atmosphere, 1 second might be all we can do, or 10 hertz might be all we can do, but it's really based on signal. So we can and we indeed do go faster as we're collecting data. We just need to add that signal up to get enough detection limit.
Oh, okay, so you don't necessarily get an equally spaced measurement in these time increments, is that
It is equally spaced
in time?
in time. We kind of are forcing it to be. So we're summing.
Yeah.
We're summing, from point A or time A to time B, and forcing it to be a second long measurement. So it's cumulative signal the detector sees across 1 second time period.
So if you didn't see Stuart's glad that you didn't find a picture.
I thought you might say that, Stuart.
Thanks, Patrick. I really enjoyed your talk. The slide with the large changes in sulfate and SO2 in an Earth system model, I think, was really interesting. I imagine putting HPTMF into a mechanism in a chemistry model is something people are, have already done or actively working on. But I think the Earth system models that are used for, say, climate projection use a much more simplified sulfate implementation. So how important is it that this makes it into those models, and what's sort of the timing or status of that?
Yeah, I mean, so this is some of the first modeling that's been done on this. There's been a lot of people who have attacked this problem beyond just Gordon's work here. I just didn't have time to go through all this. But you're right. I mean, it is difficult to put chemistry in any large-scale climate model. And so it is almost by necessity that you take DMS and you give it this very simplistic chemistry that is here on the top right. Product A goes the 2 products and you're done, and you don't have to worry about any of the inputs on that chemistry. This figure and a lot of the subsequent work is pointing out that, boy, this is a pretty large lever on sulfate in the remote atmosphere. And so, as we start to tackle the impacts of climate, as we start to say, hey, marine aerosols that contribute to cloud formation over the open ocean are one of the largest uncertainties to climate models. We need to incorporate this into those models. I don't think you've gotten very different answers in any of the modeling that's been done than what you see here. It's all basically saying that the sulfur pathways from DMS to sulfate matter. You can't do that. I mean, computers just won't allow it, but we are getting there. Jan Kazil, who's a co-PI on this project, we've talked a lot about LES modeling. Ideally, you'd put the chemistry in an LES model. You'd understand how sulfur gets out of the boundary layer in the first place, what gets removed in that process, where does the chemistry happen, and what is the impact on sulfate formation. We can start to do that, but we can't really put that in a global climate model. And so how do you, this is one of the questions we're all struggling with, is how do you model on different scales and ensemble that to get some answer? But these are challenges that need to be tackled not only for just our general understanding of climate, and this is, paleoclimate as well as modern climate. We're talking now about perturbing that system, perturbing the marine cloud system, perturbing the stratospheric system. And so marine cloud system, we perturbed that, we perturbed this. So we really do need to think about this chemistry as we think about those things. But we're all kind of struggling with the same problem, which is it's too complex to put in a model. Until computers get better.
Very interesting talk. Thank you. Have you, or would it be a good idea, have you ever run this behind a CVI to look at the what's in the condensed phase?
No. To my knowledge, we haven't run a CIMS, or nobody has run a CIMS behind a CVI. But now in my new role, sitting there working with the CVI and Adriana Bailey on the water instrument, I've thought about this about 20 times in the last few weeks. I'm looking at some data from ATOM where, boy, if we had a CVI, I might have the answer to my question already. And so it's a really great idea.
So Patrick, we have a quick question online from Minghui. Great talk. Could you comment on the application of the CIMS instrument to high latitudinal measurements in the UTLS?
I guess I'm not entirely sure what specifically you'd like to comment on, but I mean, we're starting to be able to measure in the UTLS with these instruments. This TOF-CIMS being on the DC-8 kind of got us there, but the Strat-CIMS on the SABRE mission is certainly going to be able to access that region. We're only just now developing that instrument and understanding some of the issues with it. And so as time goes on, I expect that measurement to get much improved. We not only, halogens were the low-hanging fruit. We knew we had an instrument that was sensitive enough, stable enough because of the work that we did, that we can get halogen measurements in the UTLS and specifically lower stratosphere. Organics are the next step, maybe ambient ions, things like that. So these instruments are really adaptable. Projects like this where we can now know we can put these CIMS in the upper atmosphere. We're not the only ones doing that, but I expect we'll learn a lot about the UTLS in the coming years through this instrumentation.
Thanks, Patrick. We are at the end of our hour, so if you do need to run, feel free, but I think I did see one more hand up. Was that you?
Yeah, so very interesting talk scientifically, Patrick, but you also did a really nice job in helping us understand, the role of this mundane instrument development aspect of it. And you come to it from that approach, but obviously that paid off hugely scientifically. So, as we here at NCAR and elsewhere look toward doing actionable science, how do we balance this doing the mundane and really just being driven by what can we do, what can we measure, and really responding to those bigger science questions?
Yeah, I mean, this is a really good question. I think as I've fallen into a role at RAF that's an instrument group, how does innovation, what role does innovation play in that? How do we pick the next thing to do, and is it going to pay off? 60 instruments is a lot of instruments. This is a group of, someday it'll be eight of us, hopefully again. We have to continue to support wind measurements, particle number concentrations, things like that. And we all have individual expertise out there that doesn't necessarily couple with each other. So what do you do with that, and how do you innovate with that? And I think one of the ways that you do that is by partnering across NCAR and finding, Mike Reeves does aerosols. Who are the aerosol people at ACOM? Who are the aerosol people across NCAR that Mike Reeves can couple and pair with? And we don't have to be in NCAR. We can go outside of NCAR to do that. Mike can't spend all his time doing that because he's got to operate the instrumentation in-house. And so you almost need to develop collaborations to do the kind of work that I'm talking about here, to develop instrumentation, to take that risk and put time into it. But I think that can be a mutually beneficial endeavor to do that. It can allow someone to gain resources from the RAF group or an NCAR group to help them develop a new instrument, take their ground technology and make it an aircraft instrument. And if that pans out, we can deploy it as a community resource. And so I think it does, I put in the slide, leveraging. Leveraging is a big word. We kind of have to leverage our resources around us and not be so singular in our research, especially at RAF where nobody else is doing CIMS work. But I can reach out to Kyle and Brett Palm at ACOM and work with them to help develop new technology that might then pay off in the future for our users. But I think we are limited in resources where you cannot do the development I just talked about as one person in RAF without giving up your responsibilities to do 60 instruments. So it's a tightrope.
Great discussion. Thanks, Patrick. Let's thank Patrick one more time for his great talk.